So even though we don't have, we're not going over the warm-up, I still encourage you all to do every warm-up, every exit ticket. Some of you are really good at doing that, and you probably, but we do have some students in the class who have done every warm-up and exit ticket all year, which is excellent. But I just want to remind you that these are great tools to use in order to prepare for the exams. Our next unit exam will be the midterm, which means that it should be a week from Tuesday in this class will be our midterm if my schedule matches up with what I think the school is. Yes, the warm-ups and exit tickets are a grade. If you look in Power School, you'll see an assignment that says Unit 4, warm-ups and exit tickets. And each day, the warm-up is worth 2 points and the exit ticket is worth 2 points. And so each day is worth 4 points total. If we've got 7 days, then that's 28 points. So it's not a big assignment. They're not worth many points, but it's probably, they probably are some of the most important point for effort assignment that you'll have this year. So they don't take much effort. They're not, I mean, what am I trying to say? They give you a lot of benefits, even though they aren't worth many points and they don't take as much effort. But the benefits are that you get to see literally what kind of questions you will be asked on the test. And in some cases, you see the exact test questions because I take them from the same source. So they might not be worth many points and they don't take long, just 5 or 6 minutes, but they can go a long way in terms of improving your performance on the assignments that do count a lot. That was a long way of saying, yes, they're worth a grade. But the objective today is to explain how DNA and RNA code for proteins and determine traits. The essential question, how does genetic information in the nuclei of eukaryotic cells get to their ribosomes, which are in the cytoplasm? So there's some travel involved in this process. It's a pretty complicated process, but the standards for this course don't really require that we go into all that much detail. But we should at least be aware of the fact that there's this idea of the central dogma of biology, which we'll define a little bit later on. And that idea allows us to move from genes to actual traits. Today is February 26th, TGIF. It's the third day of our 5th unit. Next week, we're going to spend a good amount of time reviewing the material from the entire semester. So we're going to be using some breakout rooms for the people. I think I have a few of you who are scheduled to be in person. You will be able to do some cool things in order to put together a big mind map that we can then share with your classmates. We will have some asynchronous assignments to cover Unit 5 specifically, but if you notice, I have not assigned any asynchronous assignments for Unit 5 yet. And that's intentional. I want you all to be able to use this time to review. So those of you who have missing assignments, you should be using your time to do that. It's not an opportunity to just not do anything. It's an opportunity to go back and get caught up. But starting Monday, we will have some assignments that will prepare us for the midterm exam. So just be on the lookout for that. Okay, we're going to start with the quizzes today. So I just sent the link to the chat to join the game. You should be able to click directly on that link, or you can go to joinmyquiz.com. 436-362. Okay, so I think there are 17 of you who are in this Google Meet, and there are now 13 of you who have joined the game. So we're waiting on a few more people. What do I want to do? Go. Okay, I'm going to go ahead and start the game. If you haven't been able to join yet, that's okay. Obviously, you can join while the game is in progress. Wow, that's fast. Okay, let's take about 2 more minutes. I know we still have 7 of you working, but you'll be able to continue working even if we jump over to the slides. We're doing a really good job. 80% accuracy is excellent. We will go over these questions when the lecture is done. Let's see. All right, so there's this idea of a central dogma, and this phrase central dogma can be applied to a lot of different fields of study. It essentially just means that this is the most important idea in the study of life, and it's the idea that genes can code for the proteins that become our traits, and also that the idea or that those genes get passed on as well. So, because this is the most important idea in the study of life, aka in biology, it's probably a good idea to try to write this down. Yes. So yeah, the DNA, which of course contains sections of genes, or sections of DNA comprise genes. So a gene is a section of DNA. I don't know why I'm struggling with the English language today. But your genes will eventually be transcribed to mRNA. And then that mRNA is translated to amino acids. Amino acids become proteins, and then proteins become traits, or they determine your traits. Okay, so let's say, and I don't know, hopefully you guys can see this a little bit. We've got this big section of DNA, and a section of DNA is called the gene. The process of transcription is going to take that section of DNA and transcribe it into the same language, but we're just writing it in a different format. And that format is called mRNA. So there's going to be a long section. So on and so forth. And of course, where do we find the DNA in a eukaryotic cell? Yes, it's in the nucleus. So this would all be inside of a big nucleus. And so we'll talk more about this, but the mRNA is going to be transcribed inside of the nucleus, but then it has to leave the nucleus because it eventually has to go to a ribosome. Okay. Cool. So I asked these same two questions yesterday. Hopefully you guys had an opportunity to write them down, the answers. Why does our DNA need to be replicated? Does anybody remember why?
Maintain chromosomes.
Thank you, Lisa. We've got to maintain the same number of chromosomes because there has to be a copy available for the new cells. If we're going through this process of mitosis, there has to be a copy available for 2 new cells. So we've got to copy the DNA before the cell can actually divide. Strongly encourage you all to write down the sentences and phrases that are highlighted in yellow at the very least. Okay. And the second question was, where does DNA replication occur?
In the nucleus of eukaryotes and in the cytoplasm and prokaryotes.
Good. In a eukaryotic cell, DNA replication is going to happen in the nucleus. But of course, prokaryotic cells do not have nuclei. So DNA replication has to happen in the cytoplasm. So what I just drew on the board with this nucleus, this is only representing what would happen in a eukaryotic cell. And I just want to mention the scale at which these things are taking place. So for whatever reason, the United States does not use the metric system. We use the standard system. So we're used to units like miles and feet and inches that really are kind of difficult to put into context. But most of the world uses the metric system, which means that a kilogram is 1,000 grams. A kilometer is 1,000 meters, because we use that same prefix kilo. A centimeter, 100 centimeters is a meter, and 1,000 millimeters is a meter. So we've been given this easy reference point. To put this into context, let me draw a number line here. If I've got one here, well, let me just find an image online. Now that I have a whiteboard, I just want to draw everything. Oh, I'm not even sharing my screen here. Oh, technology. Okay, so let's see. Can we see this? Yes, we can see this. So we can see these metric prefixes. We're familiar with many of them. Those of you who are interested in computers, then you probably heard the term gigabyte, which tells you that you're talking about 1 billion bytes, or terabyte, which would be 1 quadrillion bytes. But of course, these are scales that exist in a logarithmic function. So you're increasing by a factor of 10 every time you go up or every time you go down. So we can kind of imagine how small a millimeter is. In fact, most rulers have centimeters on them, and you can kind of imagine that a millimeter is pretty close in terms of width to a centimeter. But the scale at which we're talking about things is at the picometer scale. So it's 10 to the negative 12. It is extremely, extremely small. Your DNA is really long. In fact, like I said yesterday, I think it was in this class that I mentioned it. if we were to unravel the DNA from just one cell in your body, it would be about 6 feet long. So that tells you how tightly packed it is. And of course, we've got 3 trillion cells in our bodies. So if we were to take out the DNA from every single cell and stretch them out end to end, we'd be able to travel to the sun with that length. So there's a lot of DNA in your cells. I also want to put this into some context in terms of the time as well. The amount of time that this process takes is about 1/10 of a second. So from the time your DNA is replicated, and then to this point of transcription, and then to the point of translation, all of this is happening in less than a second. And it's happening multiple times at any given time. The cell is doing this for multiple proteins. So the way that we talk about these processes don't really do them justice, because they really are quite remarkable. They happen so quickly, and they happen at a scale that is almost unimaginably small. So I want to ask another question today, one that I did not ask yesterday, but it has been asked before. What is a cell's primary purpose? Does anybody remember that? What do most cells do? There could be multiple right answers for this.
What's that word name called? Reproduce or what's that word for like breathing?
Respiration. Okay, so yeah, this is. I'm glad we've come up with 4 really solid answers. Janelle said maintain homeostasis. Lisa just said respiration, and reproduction, which is really similar to what Selly said. Selly said that growing and replacing one another is one of their primary purposes as well. So I really should have phrased this question differently, I'm sure. The answer I was looking for, though, was the one that Abigail gave, which is their primary purpose is to make proteins. This applies to most of the cells in your body. Now, some cells are not so good at making proteins, but most of them are charged with doing one thing for their entire lives. They don't even really have to worry about reproducing. All they need to do is just make proteins. And those proteins will eventually become the products that make you what you are. We know that our cells are able to make thousands of different types of proteins. But we talked about 5 proteins specifically in this class. Is anybody writing anything here? No. Antibodies, enzymes, insulin, hemoglobin, and hormones. Okay, let's see, how much do we remember? What is an antibody? Does anybody remember that? What does an antibody do? Yes, so we know that it's related to this word antibiotics, because they're both related to like the immune system. So an antibody helps our cells to recognize pathogens. And then when we recognize those pathogens, we send in other types of cells to kind of kill those pathogens, if possible. Of course, the problem with new viruses and new bacteria like COVID-19 is that our bodies have not built up these antibodies, so we just don't recognize those pathogens right away. And by the time we do start to recognize it, it's too late. The virus has already started to reproduce. What about enzymes? What do enzymes do? Yeah, Lance says that they are chemical catalysts, which means that they speed things up. They speed up chemical reactions. yesterday when we talked about DNA replication, we saw just how many enzymes are involved in just that one process. So enzymes are really, really common proteins that are found throughout our cells, throughout our body's cells. What about insulin? This is probably also one we're familiar with. Yeah, so insulin itself doesn't actually do the breaking down, but it signals the breaking down. It triggers the breaking down to happen. Oh, have to pay for it? Yeah, insulin is extremely costly. Good knowledge there, Lance. Thank you. Get some insight into the sickness of the American healthcare system. Okay, and then hemoglobin. Does anybody remember what hemoglobin does? It is in the blood, that's correct. What does it do specifically for the blood? Yeah, hemoglobin carries oxygen, specifically in red blood cells. And then lastly, hormones. Hormones are the chemical messengers between cells. Hormones allow the cells in your brain to communicate with the cells in your reproductive system, or they allow the cells in your pancreas to communicate with the cells in your kidneys. And the highway on which they travel is the circulatory system. They travel in the blood. So that's the T in Sternger, transport. It's been a long week, hasn't it? I think everybody's a little tired. Look at Adina. I feel you though. There's a nap in my future. What'd you say? I'm with you on that. Okay. So we talked about DNA on Wednesday when we started this unit. And I think we did talk about RNA as well. I kind of gave you guys some specific insight into the chemical structure of DNA. But do we remember any differences between DNA and RNA? RNA has the nitrogen-based uracil, whereas DNA has, you can just give me the letter, T, thymine. What else? Do we remember anything else? RNA is single-stranded and DNA is double-stranded. And I see, Janelle, you jumped in and said that too. Thank you. That answer came from Sally. I know you guys can't hear the people out here. And there's one more important difference here. What'd you say? Yeah, so RNA, this is coming from JT, stands for ribonucleic acid. And that name tells us that their sugars are different. So the backbone of each of these strands is made up of a sugar phosphate complex. In DNA, the sugar that is used is called deoxyribose. And RNA, the sugar is called ribose. So DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose, RNA has ribose. DNA has A, T, C, and G nitrogen bases, whereas RNA has A, U, C, and G. can't tell if people are still writing or not. Okay. There are 3 types of RNA, which is also a difference between RNA and DNA. mRNA, the M stands for messenger RNA. mRNA carries the DNA message. This is mRNA. It carries the DNA message from the nucleus to the ribosome. The ribosome is actually made up of a series of proteins and a complex of proteins that interacts with rRNA. So rRNA is a specific type of ribonucleic acid that helps make up the ribosome. And then there is tRNA, transfer RNA. The job of tRNA is to bring amino acids from various places in the cell to the ribosome so that they can be used to make a new protein. So cells are really, really efficient. So when they engulf something else and they have enzymes to break it down, they don't just like get rid of everything. Not everything is a waste product. In fact, very few things are waste products. So they will use the amino acids that were at one point a part of a protein that got broken down. They will use those amino acids to make new proteins that the cell actually needs. So you'll have all these different amino acids that are kind of spread out throughout the cell. And it's the job of this tRNA to go find them and bring them to the ribosome so that they can be put into a new protein. We'll talk more about this tRNA on Tuesday when we talk about translation. Okay, let's watch this.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Alphabetically, transcription comes before translation. So it should be relatively easy for us to remember that transcription is the first process. All the transcription is to convert DNA into an mRNA message, a messenger RNA message. Of course, transcription happens wherever DNA is, just like DNA replication. So in eukaryotic organisms, we find the DNA inside of the nucleus. So that's where transcription happens. In prokaryotic organisms, which do not have nuclei, transcription is going to happen in the cytoplasm. Okay. The steps are relatively simple. The double helix is unzipped, just like it is in DNA replication. There is another enzyme. This one is called RNA polymerase. That actually makes the mRNA strand. We've got to keep in mind that RNA uses uracil instead of thymine. So the helix is unzipped, and then DNA polymerase uses it to make an mRNA strand. Then that mRNA is going to leave the nucleus. You can kind of see this whole process behind me. I did my best. I'm not an artist, but I think it works. You have a question, Lisa? Guess not. How's your head, Sally? Me? My head feels fine. Okay. So a simple analogy, transcription is pretty similar to having to decode a text message. If you use all these text abbreviations, I don't know if people really still use these. Maybe some of them, LOL, LMAO, those are pretty common. But you're decoding them, they're still in the same language. They're all in English. You just kind of need to write them slightly differently. Same thing with this process of transcription. Both DNA and RNA are written in the language of nucleotides, but we're writing them a little bit differently. DNA has thymine, it also has deoxyribose sugar. RNA, on the other hand, has uracil and ribose sugar. They're both nucleotides, but there are some important differences between them. So in your notes, I would like for you to write down these two DNA strands and tell me if you can transcribe them. You should be able to. It's pretty simple. We did a little bit of this yesterday. But of course, remember, RNA, when we're transcribing, we're going from DNA to RNA. And RNA does not have T, it has U. Okay, does anybody think they can give me the whole thing for the first one? Lance, what say you? Good. Not bad, not bad. I think we could have used you yesterday. We tried to play a game. It didn't really work out. There wasn't enough energy, I think, involved. So we'll try it in a couple of weeks when you guys are back here. Okay, who thinks they can do the second one? Go ahead. Oh, you got it? Okay, let's go. Excellent. Be good, sir? No. I always like to break it down because it confused me the first time as well. This is so pretty though. I don't want to erase it. Okay. Can you see this if I write right here? Okay, so the T, when you transcribe, becomes an A. The A, when you transcribe it, becomes a U. The reason for this, typically T becomes A and A becomes T, but there's no T in RNA. So I guess I should do it. This column is DNA. This column is RNA. A C in DNA gets transcribed to a G in RNA, and a G in DNA is transcribed to a C in RNA. Do you see how that lines up with what we did here? A is transcribed to U, T is transcribed to A U, C to G, G to C. Cool. Now this is just for transcription, because yesterday we talked about DNA replication, in which case we were just making a copy of the DNA. So T became A became T. If we were going DNA to DNA, we wouldn't have any U's. So really the only difference is this right here. Just got to be careful with that. What time is it? We've got 9 left. And I think this is it. There is an exit ticket. Ashanti, I know you. I was thinking you would have. So, what do you want to do about it? You want to start it now, and then okay. 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 Yeah. Let me look it up. It is B as in brother, IO, U4, P as in Tyrannosaurus, W, M as in mother, HS. So biology, unit 4, test, West Gothenburg High School. Okay, so like I said, there is an exit ticket, but we don't have much time to really do anything else. I look forward to seeing who's coming in next week. Brianna, no, that's my other class. Janelle, Ashley, and Lisa. That'll be fun. I think Janelle is going to be good at that game that we played, as long as you guys are willing to help out. Essentially, the way it worked was I wrote 8 or 9 board, and then they was supposed to be like describing terms without saying them. So what's that game called? Categories? No, it's not categories. Apples to apples is similar. Anyway, he couldn't say the word, and they were supposed to be able to say the words based on his description. Correct. Yeah. And it just took a long time. It took 11 minutes to get through. The problem is because you got to talk directly into the computer because this microphone is just really not good. But I think also people at home were perhaps a bit lethargic. Technology, right? Anybody doing anything fun this weekend? What you doing? What? Working? And you're doing what? Really? That's fun. Have you started already? So are you gonna start in the parking lot? Good idea. Okay, so it wasn't exactly a parking for anybody. You know? So how does it work here? Do you have driver's ed? So can you take a test when you turn 16? So do you have a learner's permit? Okay. But is it app? I'm pretty sure, is it a paper test? When I took it, of course I'm not from North Carolina, but it was a paper test. It was like this like basic piece of paper. And you just had to like mark out, there were different street signs, and you had to like write with the street signs. I don't know if it had really nothing to do with, like, there's some street signs that I still have not seen to this day. No practical use at all. Yeah. That's how you guys do most of your learning, right? YouTube. It can be. It can also be pretty dangerous too. That's a conversation for another day. I like YouTube too. It's just like any other social media. It has a lot of benefits and a lot of downsides. So, I think just as many. I think you could probably say that YouTube is more beneficial than Facebook. I think you could probably make a case. Do you guys even have Facebook accounts?
I mean, I have one, but I don't.
What you say? You just put it on close. It's not that you don't have one. Do you have one there, Seth? JP, do you have one? A Facebook profile? That's so funny. I think it's just like such a clear generational divide.
I made one.
Okay. So what's like the most popular is TikTok?
I use Instagram.
Instagram. I know some people, like last year I had students who I did like, even their number, can you text them and see, have they gotten my message? You'd be like, oh, I don't text, I just use Instagram. I just DM. It's like,
Yeah, a lot of people do that.
What about Snapchat?
That's almost it.
Really? Is it, you use it for what? To Snapchat people or do you use it? Because I know it also has like, you can watch like celebrity stories and news. Yeah, and it filters, right? That's a good idea too. What about Clubhouse? Does anybody use that? You never heard of it? It's weird. It's generational divide. Clubhouse is new though. That's why I thought you guys were. Yeah, I think it's new. I just heard about it a few months. Basically, people just get on there and you can start chat rooms essentially, but people are actually talking. And so you can like say, my topic today is going to be like the weather. And you can invite other people to come in and discuss the weather. And you can have an audience who just listen to those stories.
Like a podcast.
It's basically like an ad hoc podcast. You just come up with that on the spot. And you can let whoever you want to talk.
I've also heard of like Spoon.
Boom.
Yeah, I have no idea what it is. I've just seen ads for it like everywhere. I'm like, okay, why tell me about a podcast?
That must be nice.
There was this app that was getting advertised for a minute called Fight.
It's a social media.
I heard about it.
But But do you guys think being a social media
It depends.
Well, it depends on what your brand is for.
influencer? Well, why do you say it depends?
I don't know. Some people, they're not really influencing people. Like,
They're just like bragging about their money.
yeah.
You actually have to have some social influence too.
Yeah.
For an actual talent.
Yeah.
Well, people make real money from it, so I don't know. I guess you could consider it a job in accent. Face people.
But then like it's all based on ad revenue.
Right.
It's a nifty.
I heard that there's a saying if the service is free, if the service is free, collecting data on your telephone. This is called social media, social CMBA, that was thing. Yeah, so social, it's called the social dilemma. That's what it's called. Just how to do it. Yeah, it's good. I would definitely recommend it. It's not bad.
Anybody else use Twitter?
Twitter?
I literally just have Twitter just because it's too many places.
I know this really.
The reason why I have Twitter is to like get like promotional things for a couple games I play. Yeah, like if they're like I watch Twitch every now and then, but like not really.
Yeah, that's what I mean. Discord as well.
Yeah.
Discord?
Yeah.
It's like
It's
like an app for different communities. For
it's a better version of Zoom, I would say.
kind of, but it's like,
Somewhat in a way.
I've never heard of it.
Discord is like, you can talk to people